Welcome to Almost Here, Round the Corner Future Technology podcasts with Richard Jacobs. Future technologies poised to transform our lives for better or worse are the focus of this podcast. Almost here means these technologies are now here and starting to be used, or just around the corner, from Bitcoin to artificial intelligence, 3D printing, blockchain, virtual reality, and more.
Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcasts. And my guest today is Donald Richard Ort. He's a professor at Illinois University, and we're going to talk about genetic improvement of crops. So, Donald, how are you doing today?
I'm doing well.
Yeah. Well, tell me, so what is your department about, first of all, and then what does your specific research entail?
Well, I'm a faculty member in the Department of Plant Biology and also in crop sciences. And these two departments are engaged in a range of research, from ecology through to systematics and taxonomy, physiology, and to genomics. My particular area of research and my expertise is in the area of photosynthesis, and a significant part of my research program is to work on reengineering photosynthesis to be adapted for global climate change, as well as to improve the efficiency of photosynthesis in agricultural situations.
Well, I guess, I've taken biology in college and all that, but legend has it photosynthesis is like super complicated and has hundreds of reactions. Is that the case? Or, what can you tell the layperson about it that's impressive?
Yeah,
the process seems to be very impressive.
And so the process from the absorption of light to the synthesis of sugar is pretty complicated. It contains about 180 processes or reactions. The thing about photosynthesis is that it's the best understood process in all of plant biology. And so we understand who all the players are, all the enzymes that are involved and all the reactions that are involved. We also understand a great deal about the regulation of those enzymes. And so what this allows us to do is to create in silico models. And so we can take every one of those 170 processes and model it in a computer. And these are mechanistic models in the sense that we can put in kinetic information to really simulate what's going to happen. And what's powerful about these models is we can then begin to ask questions if we want to optimize photosynthesis for a particular situation, say the increase in the CO2 concentration that's already in the atmosphere. We can then begin to query these models and ask, what are the kinds of things that we would change in order to optimize it? And as you already observed, this is incredibly complex, and that makes it unintuitive. And so it's unintuitive that if you change this, what that's going to do at the level of the plant in the field and how that's going to impact yield. And so these models are able to identify targets for us. And once those targets are identified, then we can go in and do the biology, go in and do the genetic engineering, and see if the model's right. And often it is.
Well, I don't know if this is true or not. I've spoken to a few people that deal with photosynthesis. I had heard, I don't know, maybe I'm mistaken, but the average photosynthetic efficiency of most plants is, 1%? And the superstars of the world are, 3%?
Yeah, I mean, it's a little bit... it's a little misleading from the standpoint that if you start with the entire solar spectrum, about 50% of that is outside of the visible range, outside of the range that we can see, and plants can't use that light either, and so 50% of it's thrown away right away. And so if you redo that calculation based on the photosynthetically active radiation, which is the visible spectrum, you can double all those numbers. But yes, that's true that on average for a plant in the field, it's converting on the order of 1% of solar energy into carbohydrate or 2% of visible light energy into carbohydrate. And there's lots of reasons for that. Okay, go on.
Yeah, what's interesting, a couple of things came to mind as you were talking. So you said now we understand all the reactions that are going on.
Mm-hmm.
So question one, can we do photosynthesis without a plant's help? Can we do it in the lab? And if not, what's missing to be able to do that? And then, I know you have your optimization work next, but.
Mm-hmm. Well, there certainly is efforts to do artificial photosynthesis, and some of this is done by what's called biomimicry, and that is trying to mimic how the biology does photosynthesis. And then there's another effort in artificial photosynthesis to really design completely new ways to capture light energy and convert it into either another kind of energy that's usable or a compound that's usable. The fact that we understand the reactions and how the particular enzymes work doesn't mean that we can recreate them. And so some of the complexes within photosynthesis that do the real magic, the initial conversion of light energy into what's called a charge separation. These are very complex solid state multi-subunit proteins that we know the complete crystal structure of now at the atomic level, and you can manipulate them. But it turns out that they're extremely sensitive to structure. And so even small amino acid changes, the vast majority of these don't improve. They're to the detriment of the efficiency. And so re-engineering some of these complexes, doesn't mean it's never going to happen, but it's been unsuccessful so far.
And you're looking to optimize the process. The other thing that came to mind is how many different wavelengths of light are the candidates for use in photosynthesis, and are there higher energy parts of the spectrum that could be used to do a photosynthesis that, again, has a different input?
Yeah, so as I mentioned earlier, it's the visible spectrum, and so it goes from the blue, which is about 400 nanometers of the wavelength, up through the red, up to about 700 nanometers. And so this is the range that we can see. And so below the blue is the ultraviolet, ultraviolet B and ultraviolet A. The energy in this part of the spectrum is fairly low because a lot of it gets filtered out in the stratosphere. And then above that, above 700, there's all of the infrared, and so there's a lot of energy out there, but the energy per photon is fairly low. And so much beyond 700 nanometers, there isn't enough energy in a photon to carry out the basic reactions of photosynthesis.
So what does it tell you that nature has settled in on the wavelengths that are in use right now? Do you think it's found a sweet spot, or do you think there's a lot of room for optimization?
I think that's a really good question. And so, whereas I just told you that photosynthesis only operates from 400 to 700, that's true of plant photosynthesis. And those are, that's sometimes called oxygenic photosynthesis. So those that have the ability to oxidize water to make oxygen. And so that's where the system gets the reducing equivalents, or the electrons, to do net photosynthesis and net carbohydrate synthesis. But there are photosynthetic bacteria that are anoxygenic, and so they're not able to use water as an electron donor because they're not energetic enough to oxidize it. And some of those operate in the infrared, some of them out to 850 nanometers. And so, what evolution did in plants was to take two very similar reaction centers, one that's able to oxidize water, one that's able to reduce a compound called NAD, and together they're able to do the complete reaction of photosynthesis. The thing that that's not the way you design a solar cell. And so if you wanted a two-junction solar cell, you would select non-overlapping absorption bands. And so in the case of photosynthesis, these two reaction centers, which are like the two band gaps that you have in a solar cell use the same wavelengths of light. And so that automatically divides the efficiency by 2. If evolution had taken, the reaction center from plants that's able to oxidize water and coupled it with a reaction center from photosynthetic bacteria that operates in the infrared, then in principle you'd have a true 2 band gap solar cell, and you wouldn't be dividing the efficiency by 2 because they're using different parts of the spectrum. Evolution didn't do that, but it is one of the kinds of things that we think about from the standpoint of engineering. Both of these things exist in biology. Is it possible to couple them together into one membrane? Go on.
What's your overall impression of photosynthesis? You think that, again, nature did an amazing job and it did the best it could, and there's, tons of room for improvement? Or do you think that it just did a mediocre job and, it's low-hanging fruit in order to be able to optimize?
Yeah. I mean, photosynthesis is an amazing process, and and it's remarkable it evolved. And of course, if it didn't, there wouldn't be any life on the planet because it supports everything. Did it do a mediocre job? Well, in the area that it did a mediocre job, I would say is one of the major enzymes in photosynthesis is an enzyme called rubisco. And it's the enzyme that actually captures the CO2, gaseous CO2 out of the atmosphere and puts it onto a sugar molecule to make a 5-carbon sugar into a 6-carbon sugar. And that enzyme evolved, 3.5 billion years ago or so. And it evolved in an atmosphere that had no oxygen. And so photosynthesis oxygenated the atmosphere, and it evolved when it wasn't there. And so once the atmosphere was oxygenated, lo and behold, the enzyme discovered that it couldn't really distinguish between CO2 and oxygen very well. And so in our current atmospheres, about every 3rd to 4th catalytic turnover of the enzyme, it fixes an oxygen rather than a CO2. And this is actually anti-photosynthesis. And so, this is a situation where I think photosynthesis did a pretty mediocre job. In other cases, the things that we're trying to improve are to adapt photosynthesis to an agricultural situation versus what evolution selected for, which was for good competitors. And so, if you're a good competitor and you want to exist into future generations, what you'd like to do is to sequester as many of the resources that are available, whether you can use them efficiently or not, in order to keep them from your competitors. And so, for examples, trees grow tall in order to shade out things that are under them. By growing tall, they're able to do that. They actually capture more light that they can use efficiently. And they'll only grow as tall as they can to the point where putting on all that additional non-photosynthetic material is a lesser cost than competing with the other kinds of plants. And so an example of this is that, as you know, leaves are very green, and a single leaf absorbs approximately 90% of the solar radiation that's available to it. And so if you think about a forest, then those trees are very effectively competing out everything that's below them. But the paradox is that those leaves at the top of the forest, or the top of a soybean canopy, or the top of a wheat canopy, they are getting about 5 times more light energy than they can actually use for photosynthesis. And the paradox is that one leaf layer down, those leaves are light starved. And so what we'd like in an agricultural field is not good competitors, we'd like good neighbors. And so rather than having soybean leaves that are absorbing 90% of the light at the top of the canopy, and then having all the leaves in the canopy below that being light starved, we'd really like a situation where there's more light penetrating into the canopy where it can be used more efficiently. And so one of the things that we're working on is to change that. And so you can change that in a canopy by either changing leaf architecture, changing the position of the leaf or the shape of the leaf so more light gets into the canopy, or to lower the total amount of chlorophyll. And so we're engineering plants with much less chlorophyll. Our models suggest that we would get the highest light use efficiency if the chlorophyll content of leaves were reduced by 70%. So that's a lot. And so we have soybean plants now and rice plants now that look like houseplants, and they're quite light green. And so it's kind of the thing that a farmer would hate, because to farmers and crop breeders, light green means a sick plant, and usually it is a sick plant because it's not able to metabolize nitrogen for some reason, or there's lots of bad reasons to be light green. But what we've done is
reducing the chlorophyll improve the efficiency. What's the
So imagine that we have a soybean plant, and let's say it has 5 leaf layers, and so the top leaf layer gets 90% of the light,
right.
and it's 4 times more than it can use. The next leaf layer down gets 10%, and it's light starved. The one below that gets 1%. It's barely doing photosynthesis, and the one below that gets 1/10 of 1%, and it's actually doing more respiration than it is photosynthesis. Now let's take the situation at the top of the canopy that the leaf only absorbs 30% of the light. So that one's light saturated. The next leaf layer down is light saturated. The next leaf layer down is not light saturated, but it's doing significant photosynthesis. And even the 4th and 5th layer are doing significant photosynthesis. And so that plant would not be a good competitor. If you put it with full green plants, it would lose out. But in a monoculture where everybody's like that, it's going to maximize light use efficiency across the field.
But that says the light passes through the leaf if it's not captured. I thought I guess I just thought that it gets blocked by it. So if it doesn't capture it at that point, it's lost anyway.
And so there's 3 fates of light. It can either be absorbed, it can be reflected, or it can be transmitted. And so if we lower the absorption from 90% to 30%, then there's going to be more transmission deeper into the canopy to give another leaf, and there's also going to be more reflection. And so there will be some loss of available light being reflected out of the canopy. And so our modeling suggests that there is a sweet spot, and that sweet spot is about a 70% reduction.
Huh.
So
Interesting.
There is a caveat to this, and the caveat is that while light green is good mid-season, when you have a thick canopy, it's not good early in the season. And so early in the season, if the plant is light green and it lets light through, all it does is hit the soil, and it's lost. And so our models suggest that what we'd like is to have full green earlier in the season, and then as the canopy builds up, at the top of the canopy gets lighter and lighter green. And so right now we're just making it light green throughout its life cycle, but certainly there's the potential to engineer it in a way that we turn on the light green trait later in development.
As I was going to say, are there any epigenetic triggers that would change the amount of chlorophyll in a leaf over time, and could those be accomplished by exposing the plants to a certain kind of light, changing their light cycle? I mean, I don't know, whatever.
Yeah, and so
you can change.
Yeah, and so I don't think they'd be epigenetic; they'd be genetic. And so there are various kinds of promoters that respond to different colors of light. And so plants respond to shade, and so when they're in shade, they tend to elongate, and so you get this very elongated phenotype, and that's controlled by the ratio of red to far red. And so it's the phytochrome response. And so certainly one of the things we're thinking about is co-opting that phytochrome response, because as you go down further and further in the canopy, the red to far red ratio changes because you're taking out more of the red, and it's getting enriched in far red. And so and so the possibility would be then to co-opt that to change chlorophyll concentration as a function of depth in the canopy. There's a couple of other promoters that that are, that also detect light, but they detect the intensity of light rather than the color of the light. And so, yeah,
there's probably people that are working on mechanical ways, to like you said, to have the leaves appear at different angles, so as not to obscure each other. I know indoor you can control conditions a lot more than outdoor, but you're focusing on the genetic engineering of the plants to make these changes and not other changes.
Well, and so in the case of leaf angle, particularly in the grasses, there's quite a bit known genetically about what determines leaf angle. And so, if you look at modern maize crops and modern rice crops, they have a much more upright leaf angle than did those crops, 3 or 4 decades ago, much less their non-crop progenitors. And so breeders have selected for this more upright leaf angle in grasses. And so that improves light penetration into the canopy and prevents some of the shading. The other thing that's happened, however, is that it's also given the opportunity to plant at high densities. And so what's happened with maize is that as the architecture's changed, they've been able to plant higher and higher densities. And so this is what's really driving yield gain in maize over the last couple of decades. And so while it created the opportunity to get more light in the canopy, a lot of that benefit was taken away, by planting at higher densities, which also had a big benefit.
You mentioned at the surface of the leaf where it absorbs the light, and it absorbs CO2. That's where the photosynthesis, I guess, essentially begins when the inputs come in. You said every third or fourth. I forget what you call it. Every third or fourth cycle.
Every third or fourth catalytic turner, or the enzyme rubisco.
Yeah. So it'll take in oxygen instead. What's happening at the leaf itself? I thought the underside of the leaf, I guess the stomata open and they release what? Like what else is happening in the vicinity of the leaf as photosynthesis is occurring?
Yeah, good question. Let me clarify one other thing first, and that is that this oxygenation reaction happens in plants like soybean and wheat and what we call C3 plants, but it doesn't happen in corn. And so corn has the ability to concentrate CO2 around the rubisco enzyme, and in so doing prevent the oxygenation reaction. And so it doesn't do it, but concentrating that CO2 around the enzyme also costs energy. But the corn and sugarcane and sorghum, which are the three crops that are able to do this, are more efficient than the rest of the crops because they don't do this oxygenation reaction.
Okay.
Back to your question.
The plants that do the C3, right? What happens there?
So back to your question about the C3, and so depending on the plant, they can have stomates on the bottom of the leaf, the top of the leaf, or both bottom and top. And so most crop plants they're either under the leaf or on both sides of the leaf. And the leaf is covered with wax, and so the only way that there's significant gas exchange, whether it's CO2 or oxygen or water vapor, in and out of the leaf is through these stomatal pores. And so these pores are necessary to allow CO2 to get into the leaf, but at the same time CO2 is getting into the leaf, water is being lost from the leaf. And so, typically that's a huge imbalance. And so in a soybean plant, the plant gains about 1 CO2 molecule for every 500 molecules of water that it's lost. And so and so that's an exchange that's, where you're giving up a lot of water to take up a lot of CO2. It's important to understand however, that evaporation of that water from the leaf is part of what cools the leaf. And so remember, it's taking in a lot of light energy, and a lot of that light energy is heating the leaf. And so the leaf does need to cool, and this evaporative cooling by this water loss is part of the way that it does it.
Okay, got it. All right, so you're looking to modulate again the percentage of chlorophyll in leaves so that more of the right light is transmitted through to the lower leaves. What about changing the CO2 concentration locally? Is that even possible, and outside, or it can only be done indoors?
Well, I'm not sure. And so are you saying changing the CO2 concentration within the leaf or or outside the leaf?
Outside the leaf, just locally, yeah. Is there any way that you can do that in an outside environment where you increase the CO2 concentration locally?
Unfortunately, it's happening. And so, our crop plants evolved in an atmosphere that had about 220 parts per million CO2. At the beginning of the Industrial Revolution, about 150 years ago, the CO2 concentration in the atmosphere was about 270. And over that time, to 150 years ago until today, it's gone from 270 to about 405. And so this is driven by, largely the combustion of fossil fuels. And so the CO2 concentration is going up in the atmosphere, and that in and of itself is a good thing for photosynthesis because it's stimulating the rate of photosynthesis and repressing the rate of this oxygenation reaction. But as you're well aware, there are other consequences to CO2 going up in the atmosphere because it's the major greenhouse gas that's in the atmosphere. And so it's driving warming in the atmosphere about 2 degrees across terrestrial regions of the world compared to the beginning of the Industrial Revolution. And the projections of the Intergovernmental Panel on Climate Change, is predicting 3 degrees by mid-century and as much as 5 degrees by the end of this century. And so that has, lots and lots of consequences, most of which are not good for agricultural production.
Well, what happens when plants are exposed to higher CO2 levels? I know I've seen articles on this in the past. I just can't recall at this time. But what have you observed, and is there any way to to help accomplish that in an outside environment somehow? Maybe using, I don't know, maybe scrubbing CO2 from the air and then blowing it through a field where it would temporarily raise the CO2 level and then it would go back out into the atmosphere anyway?
Well, and so I think that the thing that we're thinking about more is how do we raise, for a given atmospheric CO2 concentration, how do we raise the CO2 concentration around the enzyme rubisco within the leaf? Because that's what we can actually potentially control. And so because oxygen and CO2 are competitive for the catalytic site of the enzyme, we can raise the CO2 concentration and repress the oxygenation reaction, plus stimulate the carboxylation reaction. And so I already mentioned that C4 plants like corn do this by concentrating CO2 around the enzyme by a very complicated mechanism that includes a big anatomical and morphological change in the leaf. But there are other ways to concentrate CO2 that nature has discovered. And so in photosynthetic bacteria, both oxygenic and non-oxygenic ones, a lot of them have bicarbonate and CO2 transporters. And so they can transport CO2 into the leaf and concentrate it around the enzyme. The Cyanobacteria, these are the bacteria that actually are oxygenic. They have structures, microcompartments that are called carboxysomes. And inside these carboxysomes, which are all of the rubisco is housed. And so they have bicarbonate transporters that transport CO2 or transport bicarbonate out of the water, and so these are aqueous environments into the carboxysomes. In the carboxysome, along with the rubisco, is carbonic anhydrase that converts the bicarbonate into CO2, and it's right in the region of the rubisco enzyme. And so this is very effective in cyanobacteria to prevent this oxygenation reaction and to stimulate photosynthesis. And so part of our project, and so we have this multi-institutional project to re-engineer photosynthesis, and one of our partners at Australian National University is in the process of putting these carboxysome structures into higher plant chloroplasts. And they've been able now to get the structure in, form these microcompartments, and the microcompartments have the CO2 inside, or have the rubisco inside of them. Now they're trying to put the carbonic anhydrase in with it. What they have not yet been successful with is to put the bicarbonate transporters in. We thought this would be the easy part. And while they can get the carbonic, or they can get the bicarbonate transporters into the membrane, for reasons that are not understood at this time, they're not active. And so we have to figure out how to activate them. And so it's interesting, because these cyanobacteria are the progenitors of the chloroplast in higher plants. And so
supposedly that's what happened. It was like a symbiogenesis where the blue-green algae merged with the plant cell, and that's what became the chloroplast.
Yeah, and so one of the questions is, did that happen, prior to cyanobacteria having these carboxysomes, or did they have the carboxysomes that moved into plants and then they lost them for some reason? It looks like the phylogenetic record suggests that the endosymbiosis of the cyanobacteria into the higher plant happened prior to the formation of carboxysomes in these organisms. So it's really attractive because, the modeling suggests that this could be over a 50% improvement in photosynthetic efficiency if we could get them in and functional in higher plants.
Yeah, it's amazing. I mean, I was going to ask you if there's even more ways you're looking at to improve the efficiency, but I get the feeling you have so much work ahead of you just with these two methods that you don't need any more.
Well, let me tell you another one that my group is working on, and so I told you about this oxygenation reaction. And so once this oxygenation reaction occurs, it forms a molecule called glycolate, and it's just a 2-carbon compound. And because it's a 2-carbon instead of a 3-carbon compound, it can't be put back into photosynthesis. On top of that, it's toxic, and so the plant has to deal with it. And the way the plants deal with it is a process called photorespiration, which is very complicated. It involves 3 organelles, 25 or 30 reactions, and uses a huge amount of energy. And so one of the things that we're doing in our group is trying to engineer a more efficient way to metabolize that glycolate than the native pathway does it. And so, for instance, E. coli have to metabolize glycolate, and they do it with just 5 gene products, with just 5 enzymes, and it doesn't use nearly as much energy as the plant pathway. And so what we've done is we've taken that E. coli pathway as well as 2 other synthetic pathways that we've designed and put those into the chloroplasts of higher plants. And then we've also genetically inactivated the exporter of the glycolate. And so the glycolate can't get out of the chloroplast into the native pathway and is forced to go through the pathway that we've put in. And so our model suggested that this would be a more efficient pathway, and it turns out to be. And so our model crop that we've been doing this in is tobacco, and we've seen as much as 40% gains in biomass grown in the field with these photorespiratory bypasses installed. And so this work was just accepted in Science last week, and so it'll be coming out, sometime in the next few weeks.
That's great. Wow. So what are your targets? I spoke to one researcher whose target is to improve the yield of rice, 10% or so. What are some big targets that you have that you're looking at through all this research?
Well, I think what can be said is that photosynthesis is pretty highly conserved, and so there's this dimorphism between C3 and C4, but if you look at C3 plants, they do photosynthesis. They all do it the same way. If you look at C4 plants, they all do it the same way. And so these kinds of things that we are discovering that are working in tobacco should work in, rice, and they should work in wheat, and they should work in soybean. And so that's the advantage of, this conservation of mechanism. The disadvantage is that there isn't much diversity out there for breeders to select for. And so, nearly all of this is going to have to be done by engineering. And so the kinds of things that we're seeing in tobacco right now with biomass increases of, 30 and 40% for some of these, as these go into food crops, which are more difficult to work with, and we start looking at seed yield rather than just total biomass, it's undoubtedly going to be the case that we're not going to be seeing 40% increases in soybean yield. But certainly, I think that 20% increases in soybean yield is a viable target, and we think that because we're suppressing this process of photorespiration with these synthetic pathways. If we suppress photorespiration by just growing plants at elevated CO2, we do see these 20% increases in seed yield. And so we think doing it genetically versus doing it by increasing the CO2 in the atmosphere should give us pretty much the same results. Of course, we don't know that until we get into the soybean, but that's what we're doing now. And so we're taking these traits that we've engineered in tobacco, and we're starting to engineer them into soybean, cowpea, and cassava.
Well, last couple of questions. What do you see that climate change is doing anyway to all our plants and our foodstuffs, and what's happening now? What's going to happen over the next 10 or 20 years, regardless of what we do?
Well, I don't know, regardless of what we're going to do, but certainly the work that we're doing is to adapt plants to the climate change that we think is coming. Here at the University of Illinois, we have a global change research facility known as SoyFace. We're able to raise the CO2 concentration in the field for the entire growing season to levels projected for 2050, 2060, or 2070. And at the same time, we can change canopy temperatures using very well-controlled infrared heaters. And so we can experimentally ask the question, if we just raise the CO2 concentration, what happens? What if we raise the CO2 concentration to the level predicted for 2050 and we raise the temperature to the amount predicted for 2050? And so we can begin to ask these questions. And so CO2 alone, as I mentioned earlier, really drives yield. Temperature alone really suppresses yield, and temperature by CO2, increasing both of them, the elevation in temperature pretty much negates the stimulation that you get from increase in CO2. And so there may be ways to adapt plants to do that. We think that there are, and so we're working in those candidates. The other thing that's happening with global climate change is you put more energy into the atmosphere, which is what warming is. It changes precipitation patterns. And so for the Midwest, the Corn Belt, which produces 40% of the soybean and 40% of the maize in the world, we don't expect much change in total seasonal precipitation. But the amount that occurs during the winter versus during the growing season is going to increase. And the intensity of the rain events is going to increase, which means that the duration between rain events is going to increase as well. And so, it's possible to get, the same 1,200 millimeters of rain and not have it benefit the crop nearly as much because too much of it's outside of the growing season. And even when we get rain events during the growing season, they're spaced far enough apart that we'll have drought between the events. And when the events come, not all that water will be absorbed and we'll have more runoff. And so, and so what this says is that, we have to adapt crops, genetically to do things differently. But we'll probably, we certainly have to do management changes as well in terms of when we plant the crops and how we plant the crops. And while our research doesn't involve, the mitigation of climate change by reducing the production of greenhouse gases, that has to be part of the equation if we're going to feed 11 billion people in 2100, or even 9.5 billion in 2050.
Well, we're just about out of time. I really appreciate all your knowledge and information. It's really fascinating. There's so much going on, so much to work on, and huge challenges ahead.
There are huge challenges. There are huge challenges. I think many of us working on it are optimistic. I think that there's lots of things that we can do. And, as I already said, we're working on one part of it. We're working on the adaptation part, and we have faith that the people that are working on the mitigation part, that those two things together, that we will meet this challenge, maybe not in the way that we're envisioning right now, but in some fashion that's acceptable to humanity and to the planet.
Well, very good. So resources for listeners, how can they find out more or maybe get in touch with the lab, see what you're doing in more detail?
Well, we have a couple of websites, and so there's a website for our project called RIPE, which is an acronym for Realizing Increased Photosynthetic Efficiency, and so you could Google RIPE. And then we also have a website for our global change research facility called SoyFace, so soy as in soybean, and FACE is an acronym for free air carbon enrichment. And so if you Google SoyFace, you will find out what we're doing there. And in both cases, there's a lot of background information and source references and contact information.
Okay. Well, very good. Well, I appreciate you coming on the podcast and spending your time. Thank you.
Well, thanks for your interest. Bye.
You have been listening to Almost Here, Around the Corner Future Technology podcast with Richard Jacobs. Subscribe to this podcast, post a review to discover more future technologies that are poised to transform our lives for better or worse, such as Bitcoin, artificial intelligence, 3D printing, blockchain, virtual reality, and more.